So for those of you who don't know, there is quite a big program looking at the fisheries in Flagboshello and Glen Alpine Dams, I think run by Mandler and Albert and the rest of the freshwater team. Phumza is helping with that program. She's looking at the biology of 6 of the fish species in Glen Alpine. And that's all I'm going to say. Everybody knows Phumza? Welcome.
Morning. Thank you for coming to my talk. My title is The Role of Morphology in Structuring the Feeding Niches of the Dominant Fish Species that are inhabiting the Glen Alpine Dam, which is within the Limpopo River system. Firstly, the feeding morphology plays an important role in promoting in facilitating resource piping and enabling species coexistence through niche differentiation. Because the feeding performance of the fish is determined by morphological characteristics, which influences its ability to locate prey, to chase prey, to capture prey, to process prey, and also to digest the prey. And then several key functional feeding traits, such as the gut length, the tooth shape, the mouth width, and the mouth orientation, body depth, have been identified as strong predictors. For example, the gut length, for example, if a fish has a long gut, that means that that fish is more likely to feed on prey that are hard to digest, such as plants and the detritus. And then if a fish has mouth width has something to do with the size of prey that enters the mouth, and then the tooth shape have something to do with the amount of deformation after chewing. And then other examples you can think of, the thickness of the pharyngeal jaws. It explains, for example, if a fish has thick pharyngeal jaws, that means that that fish is feeding on prey that are hard to digest, like snails and seeds. And then because of this difference, because of the relationship between morphology and the functioning of fish during feeding, feeding morphology structures can be used to predict food competition between species. But for the purpose of the study, I'm going to focus on the Oreochromis mozambicus, the Coptodon redali, Enteromias trimaculatus, the Macassenias crameri, the Petrocephalus wesselsi, the Shilbe depressirostris, together with the Micropterus nigricans, since they were the ones that were common in that dam. And starting with the Micropterus nigricans, that fish, starting with that one, it is native in the lower Great Lakes and the Mississippi River system, and it was introduced into South Africa in 1928 for angling. It's causing lots of problems in freshwater systems because it feeds on fish. And then under the IUCN Red List, it's being categorized as being least concern. And then we have the Oreochromis mozambicus, which is native to the lower Zambezi, southwards to the Brack River, including the Limpopo River system, which is our study area. And under the IUCN Red List, it's being categorized as being vulnerable to extinction because of due to hybridization with Oreochromis niloticus. And then we have the Coptodon rendali, which is native to tropical Africa from the lower Pongolo River to the Kunene River, including the Limpopo River system. And under IUCN Red List, this species is categorized as being least concern. And then we have these 4 species, the Petrocephalus, the Macassenias, the Shilbe, and the Enteromias trimaculatus, all are being categorized as being least concern under IUCN Red List. And we have the Petrocephalus, which is endemic to the Komati River system. The Macasenas and the Shilbe are endemic to the Limpopo River system, whereas the Trimaculatus is endemic to the Crocodile River. So this study tested the hypothesis that trophic niche differentiation is an important mechanism that facilitates the coexistence of these species in the Glen Alpine Dam. And this was done by investigating the feeding morphology and dietary composition of these species. Diet, I did it in autumn, winter, spring, and summer seasons of 2024. And then samples were collected from the Glen Alpine Dam, which is within the Mogalakwena River, which is the Mogalakwena River, which is a tributary of the Limpopo River. And this dam was constructed to supply water to the farms that are low down the stream during dry seasons. And then morphological measurements were taken following the procedures that are outlined by Nagagelke et al. 2018, whereby the external, all the measurements that are responsible for locating prey, the chasing, all the measurements that are responsible for feeding. We measured them using external body measurements, response that facilitates or that are responsible for during the swimming, the mouth measurements, together with the gills, since the gills act as sieves. And they also determine the size of prey that get retained within the fish. And then after we took those measurements, a PCA was then performed on standardized data. And then food fish model, which was developed by Sipping and Nagagelke 2001, was then used to quantify the ability of the functional feeding traits that we measured. If based on this measurement, what are the chances that this fish would be able to feed on this particular type of prey, such as the ostracods, the algae, the plants, the chironomids, the fish, the insects, and so on. And then the materials and methods for diet, where dietary measurements were done following the procedures outlined by Hyslop 1980, where the prey abundance, the frequency of occurrence, and the percentage of volume were determined. And then these values were then used to determine the index of relative importance. And then the percentage of index of relative importance was used to compare the diet among the species. And then, but then this index of relative importance is only applicable for prey items that can be able to count. Other prey items that couldn't count frequency of occurrence was also useful to explain the diet among the species. And then the diagram, just as a diagram that shows some of the stomach contents that I found on the stomachs on the diet of Micropterus nigricans, which is cichlid and a catfish. And then those are the algae that I found on the stomach of the Oreochromis mozambicus. And then these the results that I obtained. The PCA ordination represented 50% of total variation, indicating that there is a difference in the trophic morphology of the study species. And then if you look at the graph there, you can see that the two cichlids are overlapping together, meaning that there's a probability, there are high chances that they will eat similar food resources, and these individuals are characterized by long guts, long body depth, and also large eyes. And this means that these two species there are more chances that they are going to feed on prey that takes some time to digest, such as plants and the detritus. And then the large eyes has something to do with searching for prey. It means that there is a possibility that it's going to feed on small-sized prey under turbid water, yeah. And then we have the two Mormyrids together with Trimaculatus and Shilbe also grouping there, meaning that these species are also likely to feed on similar food resources. And then looking at the Micropterus nigricans, it is sitting there on its own, and it has features that are associated with sanction feeding and feeding on larger size prey. And then this diagram there on the top, it shows the probability that each of these fish species are going to feed on the prey items listed there. So if you see here, what are the chances that this one will feed on this? That's what this graph tells us. The green, it represents the positive correlation, maroon, negative correlation. The bigger the size, it means the bigger the correlation. And then looking at this table here at the bottom, the table here at the bottom, it shows some of the, it shows the diets, the results of the stomach contents that I did in the lab for the study species. And I've also highlighted here everything that was predicted to be present here, and then it was found was also found in the diet. I've highlighted it green, and then if it was predicted to be negative, to be absent in the diet there, and it's also absent here, or it occurs in low numbers, I've highlighted that in yellow. So for the diet, let's start with the cichlids. Remember from the previous slide, I mentioned that there are probabilities that these two are going to have similar diet, and they are also going to feed on hard to digest prey and small size prey and turbid water. And then if you see here, we have the presence of mud in the diet of these two cichlids and also the ostracods. And this explains the ostracods could be the reason why maybe they have large eye sizes. But also this one, Orochromys mozambicus, also fed on plants and algae, as predicted from the previous slide. If you recall from the previous slide, I assumed that plants will also be present in the diet. There are high chances that plants will also be present in the diet of this species, explaining why then this one has plants in this diet. Also, the ability of this species to also consume on other prey items also explains why they are able to coexist within the dam. And then moving on to the Mormyrids, we've said that the Mormyrids, this one and this one, will have overlapping diet. But then from the results, I've noticed that the two Mormyrids both specialized on ostracods and the chironomids. However, the Macassanias also had high consumption of insects in its diet, explaining why maybe they are also able to exist. But then when we look at this one, it has a different diet compared to these two. But then from the food specialist, from the trophic profiles, it was predicted that this organism, the Enteromias trimaculatus, will have high incorporation of zooplankton, and it won't feed on insects. Hence, I found it had low consumption of insects in its diet. And then moving on to the shilpe, from the previous slide, it was predicted that this one will have high consumption of fish. Hence, fish were also dominant in its diet. But also the species, maybe the overlap that was found on the previous slide between these 1, 2, 3, 4 species could be due to the fact that they are all capable of feeding on insects. And then moving on to the largemouth bass, if you recall from the previous slide, I did mention that this one have characteristics that will allow it to feed on large size prey, and also the feeding based on the trophic profiles. It was also predicted that it will also have high consumption of fish. Hence, fish were present in its diet, and only for insects, this particular fish only selected the large size. It only selected the dragonflies, which have large sizes compared to the other insects that were present in the diet. Now, in conclusion, the feeding morphology is a strong predictor of diet among the study species. And then despite some overlap in the diet, clear morphological adaptations were associated with differences in prey use, supporting the idea that morphological divergence promotes trophic niche partitioning and species coexistence. And these are my acknowledgements. Thank you.
Thanks, Phumza. Any questions? Go, Jonathan.
Thanks so much, Phumza. Really nice, neat work. I didn't quite get though, which which were the actual traits that separated most those groups that you identified. And the second, so this was more related to, I think, the PCA that you showed. And the second one is when you said that there are some, when you showed the red and the green correlate, negative and positive correlation. Could that, some of the partial disagreement from your original hypothesis also be due that maybe there are different diets that the fish had in their stomachs depending on the time of the year? I don't know if that graph you had accumulated all of the diets and compared, but perhaps there may be some opportunistic choice of what praise may be available. Thank you.
Yes, you're correct. And sometimes diet can depend on the region. It can also depend on where you collected, because it depends on prey availability on the environment. Maybe if we can conduct an experiment and maybe expose the fish on those different food types and see how they respond to them, then we should be able to tell exactly. And then to your first question, the cichlids had long guts and large eyes. And then the Mormyrids had features, have long, the mouth measurements were large, and also the measurements around the gills. And then the others had small stomachs. The others that grouped there in the corner, they had small stomachs, and the body depth was also small for them.
I have a question, Phumza. So good luck on unpacking that table. That was a lot of data there. So Orychromis mozambiquus, Coptodon, and Trimaculatus are all eating quite a lot of mud. They're obviously foraging in substrate. So how can you explain the absence of ostracods and Trimaculatus?
Because maybe it's because of the size of the eyes.
Size of the eyes.
And I'm saying because maybe something that is feeding, maybe it may get muddy there.
So they're obviously feeding in the substrate, because they're all ingesting mud. And ostracods should be in the very surface layer of the mud. So I'm struggling to understand why there weren't ostracods in the Trimaculatus diet. Look there. Trimaculatus, complete absence of ostracods. And yet you've got mud through all of them.
Yeah.
Sorry, that was a tough one.
They might feed
You have to
deeper.
think about that.
They might feed deeper.
They could be feeding deeper.
Maybe.
That's possibility. All right, that's for you to think about.
Yeah, okay.
Any other questions? Yeah, yeah.
Did they have a long mouth?
No,
Not really.
All right, Phumza, maybe not to you, the people who collected the fish. Were there any frogs and tadpoles there?
From the region?
Because I remember even in your first presentation, I'm sorry.
No, not in diet. From the region, we did catch
Yes.
some frogs, but they would catch and release.
Why are there no frogs and tadpoles in the diet?
Maybe they are fast swimmers. I don't know, but I know that some fish, they also eat crabs, but only the larvae. Frogs from the stomachs that I've analyzed in the past, I don't remember the fish feeding on tadpoles.
Even fish eggs. I mean, even
Fish eggs, they do eat them.
No, I'm talking about eggs of.
yeah, so the tadpoles are going to be in shallow water, aren't they?
Yeah.
They're going to be in the top sort of meso. And probably fishing in deeper water than that, aren't you? You've been catching with gill nets.
And
Yeah,
fights
probably in deeper water.
and same.
Another thing for you to think about.
Oh,
Any more questions? Yes, yes. What are you supposed to be thinking about?
So did you buy fish from people who were fishing there? And maybe compare, because maybe they're fishing differently than how you guys will. Maybe compare from because they are. Did you not buy any fish
no, we didn't.
from people who are fishing there?
No,
And maybe compare.
no, we didn't. But even the people that are fishing there, they are using gill nets. The same gill nets that we also used.
Just a comment, so all the way through you were referring to eyesight, and I don't know whether everybody else knows, but your Marcosinius and your Petrocephalus are not feeding with eyesight, are they? They're feeding nocturnally and they're feeding with electroreception. So that also might be something you should consider when
Okay,
thinking about how they're foraging.
Okay,
Then we're certainly foraging on their heads, and the electors are producing organs in the tail, and they're probably swimming backwards and foraging in substrate like that. Quite a weird way of foraging, but it's totally different. It's not eyeball predation at all.
okay, thanks, Roger.
You guys are post festival stupid, aren't you? No more questions. Thank you very much, Phumza. Well done.